My name is Dr. Sandra Demaria. I'm a Professor of Radiation Oncology, Professor of Pathology at Weill Cornell Medicine in New York City. I have been funded actually by the Chemotherapy Foundation for 20 years. During my residency in pathology, having a background in immunology, I was fascinated by the presence of immune cells, particularly in breast cancer. I sort of formulated an hypothesis that perhaps the immune system contributes to the response of tumors to treatment with conventional cytotoxic agents such as chemotherapy. That was the early 2000s, and so there was no real immunotherapy that was available for cancer patients. And there was some resistance to the concept because everybody knew that chemotherapy and radiation therapy, which are cytotoxic modality, they can kill tumor cells, but they were also expected to really kill the immune cells. So the idea was difficult to sell. But we had some strong rationale for thinking this. And at the time, it was difficult to get funded by conventional foundations, including the NIH. And the Chemotherapy Foundation support was truly critical in helping me to jumpstart the work and really get more solid evidence for the hypothesis that over the years has become actually quite accepted in the field. And I think in general, we have learned that there is always patients that are different from others. So what works for one patient is not working for all. The support of the Chemotherapy Foundation has been really instrumental for all these years in allowing us to come up with new ideas. Very important to get a little bit of resources to allow us to test. And actually, many of these concepts that we were able to test have shown really some clinical relevance, although some of them are definitely not yet in a clinic. But we really have to constantly think of something new that can help improve responses of patients to treatment. And the support of a foundation such as the Chemotherapy Foundation has really worked as what I would like to call an engine for creativity, an engine for discovery, and for developing new directions. Most of my work also as a clinician, I'm a breast pathologist, has been focusing on breast cancer, and we definitely is a tumor that has not been considered immunogenic for a long time. And we now know that there are different types of breast cancer, that some of them actually are quite immunogenic. And for this type, particularly triple negative breast cancer, now some combination of chemotherapy and immunotherapy, with immune checkpoint inhibitors have been actually FDA approved. But we are investigating how, first of all, how to improve responses in this subtype of breast cancer, such as triple negative breast cancer. And second of all, how to make progress in the other subtypes of breast cancer, such as hormone receptor positive breast cancer, which is actually the tumor that is most frequent in patients. So the majority of patients who develop breast cancer will have this type of tumor. And we have studies both in a lab as well as in a clinic. Very enthusiastic that a lot of other institutions, a lot of other investigators across the world, I should say, there are many centers in Europe and in US that are testing similar combination, which really is kind of a measure that what started off about 20 years ago as kind of an idea that was non-mainstream has now become something that is important in the field, and we really hope we can make a difference for the patients, which is really the goal of all of this work. The concept of cancer immunotherapy is treatment will not directly target the cancer cells, but it actually targets immune cells to make them work better against the cancer. There is an immunotherapy approved by FDA for triple negative breast cancer, which occurred relatively recently, and it is a immune checkpoint inhibitors that is used with chemotherapy. And it works for the most part in patients that really lack evidence of engaging the immune system, the tumor engaging the immune system. Usually these approaches don't work. So we have been really working in trying to use radiation therapy as a mean to convert a tumor that is not recognized by the immune system in one that it is actually recognized by the immune system, where the immune cells come in, get activated specifically against the antigen of the cancer cells. This usually occurs in the lymph nodes that drain the tumor, and then these cells, these lymphocytes, once activated, they can go back and eliminate completely the tumor. So complete the job that radiation or chemotherapy or the combination of these approaches, which are often used against cancer, could not complete. The immune system can get recruited, come in, complete the job. It can also, once activated, the immune cells may also go from the tumor that was treated to the spread and actually eliminate metastasis. Usually this is not achievable when radiation is used alone against one lesion, but in combination with cancer immunotherapy, it has been actually observed. We were able actually to show some of these responses in patients. We have been trying to understand how to best leverage this ability of radiation to make the tumor more immunogenic, to convert the tumor into an in situ vaccine. And one thing that we found was that besides being taken up directly, the dendritic cells can receive messages from the tumor cells that are irradiated in a form of what are known as small extracellular vesicle. They kind of send this like almost like little messages in a bottle. In this case, the bottle is a small extracellular vesicle that can contain antigen of the tumor cells. It can also contain this material, this genetic material, this DNA that then activates the dendritic cells. So it's mimicking, in a way, a process, again, that occurs during viral infection, which is what the immune system was developed to sort of to protect us against, but can also be protecting us against cancer. And so this is a bit on a schema of we currently are working on to really understand how this initial activation stimulus induced by radiation in the cancer cells can propagate to the renal lymph nodes and to other sites in the body to carry these messages that activate the immune response. In combination with cancer immunotherapy, now we can induce responses in patients that otherwise did not have sufficient spontaneous activation of the anti-tumor immune response when treated with immune checkpoint inhibitors. This is just a picture actually from a real breast cancer in a patient. We are trying to figure out how we best can activate the good immune cells and block this counter-regulatory immunosuppressive system that try to reduce the degree of rejection of the tumor by the immune system. So I just want to briefly mention as an example another project that we are working on. We did find that radiation therapy could sensitize tumors that are otherwise unresponsive to respond to cancer immunotherapy. In particular, we have worked a lot with antibodies against a molecule known as CTLA4, which is kind of breaks the ability of T cells to kill tumor cells. So this is actually the therapeutic that was discovered by Jim Allison, who won the Nobel Prize in 2018. What he was able to demonstrate is that this is a kind of a receptor on the T cells, the lymphocytes that can eliminate tumors, that inhibits their ability to perform the effector function. And if you block it, now you can get some patients to reject their tumor. So we use this mouse model. It's a model of triple negative breast cancer. It does not respond at all if you just use the therapeutic against CTLA-4, the immunotherapy. But when you combine it with radiation, you increase survival of the mice. And this is a really very aggressive tumor. So we knew that it is effective, but just like patients, the majority of the animals are not cured. And so we went and did a really deep analysis using novel technologies that actually were not readily usable when we first started 20 years ago, but now are really available and allow us to ask more deep questions in details about what's happening in the tumor. And these blue and red sort of spots just show. The blue means there are very few good immune cells in the tumor. The more red you have the immune cells in the tumor. And this is just showing here, it's the tumor when they are not treated. When you just put the immunotherapy, there is really nothing. They are completely resistant. When you only give radiation, there is a little bit of immune cells coming into the tumor, but when you put the two together, now the tumor becomes all red. So that's a good sign. We are bringing into the tumor the cells, and this corresponds to the fact that we see responses, but try to understand why they are not as effective as we would hope, why most animals are not cured. And we use an analysis really. Each kind of dot here represents a single immune cells, a single lymphocyte in this tumor, and it's a technology called single cell analysis, which allows you to really see individually what are the T cells that are in the tumor and what is their functionality. And so out of this analysis, we got some clue of what other therapies we could use to maybe improve responses. And just to say that what we thought seemed to be the most important T cells that were associated with response, we could go back to databases of human tumor and use this kind of gene signature for the good T cells and look whether they were present, to go back and ask the question, do we have any evidence in patients that the same type of T cells may be useful in patients, not just in our animal model? And in fact, we found that there was increased survival in patients that had evidence of having this gene signature. It's indirect evidence, but it is actually supportive that this type of functionality of the T cells that we found was potentially relevant also in patients, in human beings. So then we went back to the mouse model and we asked, so this is the type of tumor regression that here is tumor going up, here is tumor keep growing after you start the treatment. And so again, there is only a subset to our combination of radiation and the antibody against CTLA-4 that go down, that regress, that would be considered responder in a clinical trial, for instance. So we came up with a number of potential targets that could be tested. And I'm just showing this to make the point that, we came up with a bunch of different targets. And none of them worked, even though our data was suggesting that perhaps this could help. Only one did work. And so this is actually highlighting the importance of using the clinical model, because you don't want to perform this study in patients. You want to try to understand what may be the best combination by doing all this work in animal model before you choose the best new treatment that should move forward to testing in patients. I really am very, very grateful for the people that do a lot of work in parallel with the scientists and the physicians that do their work in the lab and in the hospital. There are a lot of people that do this work by fundraising, by educating the public, by explaining how important it is to invest in medical research. I think that the support that we have received through the year from the Chemotherapy Foundation has really been critical and fundamental for us to test new hypotheses, to go in new directions, because you really need to think outside of the box to make progress.